pasangla, now unga Shruti ma'am and inniki we're going to revise the entire principles of inheritance and variation. So in the class or notes mattum illama, to access all our notes, notifications and updates, definitely do scan the QR code and join our WhatsApp community. Our 30-day offline crash course is now starting on March 27th and the course fee is only 10,000 rupees, and this will be very, very affordable and yet effective on a crash course for you to complete your syllabus and attempt your NEET exam. So from March 27th onwards, you have our 30-day crash course, pasangla. So definitely do visit our Coimbatore, Tirunelveli and Trichy centers because seats are very, very limited. Okay, and to get started with your crash course right now, our online crash course is starting from 26th of December and this course fee is only 4,275, and along with this, if you want the study materials and the tab as well, you can procure it at 19,574 only. So definitely do call this number and block your seat because we have very, very limited seat but many of you who want to join. So definitely do make use of this, and at our offline centers you can get a free 7-day coaching. You can come in, see the teachers, interact, attend class, see how you like it and then enroll. So definitely do go in at our offline centers, Trichy, Coimbatore and Tirunelveli. December 26th the classes are starting. So there are many doctors who have been benefited from this. So make use of this and definitely do join us. Okay, so call this number and secure your seats, everybody. So let's start with principles of inheritance and variation. So firstly, the 5 different topics, the most important topics that you need to remember is firstly Mendel's law of inheritance, what the laws are, how the monohybrid crosses, dihybrid crosses, that you definitely have to know. Then the deviations, mainly incomplete dominance and codominance. Then sex determination is again a very, very important repeated question when you look at your NEET portions. And then linkage and crossing over. Again, very applicative questions appear from this section. And then we have the chromosomal disorders, which is also definitely high weightage. Okay, so when it comes to board exams, definitely these are the questions where you can expect a lot of 4 and 5 marker questions. Okay, so firstly, talking about the laws of inheritance, we have Gregor Mendel. So he was the one who studied on the pea plant called Pisum sativum for 7 years from 1856 to 1863. So he conducted his hybridization experiments. So what is this hybridization work? I'm going to dust the pollen grain of the varieties that I want instead of letting the natural pollination happen. Okay, so why this Mendel's work is important is because he was the first to apply any mathematical logic into biology. So it took a long time for people to accept it, but nevertheless it was a very, very important discovery and important application for the scientists to know. Second thing he had a lot of samples. So large sampling size helped him deduce the laws very, very accurately. And also he verified it in other generations and only said that this is the law. It's not like he found something and he immediately saw it. He multiplied the results and verified the results very, very clearly. Okay, now then he also established the rules of inheritance as rules that will apply for mostly 99% of the cases, and it's not just an observation that he gave us. Okay, so what he chose is basically contrasting traits in the P lines. Okay, so what contrasting lines? For these 7 features, very important: height, flower color, flower position, pod shape, pod color, seed shape, seed color. So this was the 7 characters that he took, and these 7 characters had alternate contrasting characters. So tall, dwarf, there was no in between. Violet or white, there was nothing in between. So because of this, he chose true breeding plants. What are true breeding plants? How many ever times you do self-pollination, it will still show the same stable trait. That's what is called a true breeding line. Now from there, he took these 7 characters, 14 true breeding lines, and conducted his experiment. Okay, so this what traits and what are the dominant and recessive, what are the alternate forms is very important. They can ask you as a question. Okay, now from there he conducted his hybridization experiments. So his hybridization experiments included, I'm just going to give you a gist. Okay, so what he did was he chose the parents, then he removed the anthers, emasculation was done, then after that he bagged the stigma, then he dusted whatever the stigma he wanted, a proper cross-pollination was done, artificial pollination was done, and then he rebagged it so that there is no chance for any kind of contamination. Now once he did these crosses, he did a monohybrid cross and a dihybrid cross. Okay, so monohybrid cross showed a phenotypic ratio of 3:1 in the F2 generation. But in the F1 generation, he saw that only one feature was expressing itself. The other parental type was silent. It did not express itself. So that's how the law of dominance came into picture. So dominant varieties, so dominant traits are the one who will express themselves in a heterozygous condition. Okay, but in homozygous condition, the recessive one will anyway express itself, which is what happened in the F2 generation. So this was the phenotypic ratio, but genotypically it was 1:2:1. Now when he did the dihybrid cross, he saw that there was a 9:3:3:1 ratio when it came to the phenotype. But in that, because we're taking 2 genes into consideration, if you chose just one gene out of it, it was still 9 plus 3, 12, 3 plus 1, 4. So 3:4 means, 12:4 means 3:1 only. So based on all of this, he gave his first law of dominance, which means when there are 2 dissimilar alleles, only one will express itself. Then second thing he said was there is no blending. They segregate and they unite independently. And when he saw the dihybrid cross, he gave the world the third law, which is the law of independent assortment. So in that he said that irrespective of how many genes you take into consideration, one gene will assort independent of the other. So they are not related. This is all what Mendel said. Now moving to the next most important topic, which is incomplete dominance and codominance. So there are a lot of deviations, which includes incomplete dominance, multiple allelism, pleiotropism, polygenic inheritance, codominance, but of which these 2 are the most important. Okay, so in Mendel's rules, what he said was, if there is a heterozygote, okay, then only one will express itself while the other will not express itself. So phenotypic ratio was 3:1. But in the case of incomplete dominance, there is an intermediate between the 2 parents. Now there was a scientist called Carl Correns. So he studied the snapdragon or the antirrhinum species, and in that there is a red flower, there is a white flower. When you cross them, he got all pink flowers. So that means phenotypic and genotypic ratio is the same, 1:2:1, where 2 is the pink flowers instead of being red. Okay, so this difference is because one allele is not completely dominant over the other. Okay, so that is why it is called as incomplete dominance. So what this dominance actually is, it is basically a gene that codes for some product. Okay, it can be some enzyme, but mostly when it becomes a heterozygote, why is it a heterozygote? Because there is another allele that has been modified into what? Either it produces a normal or a less efficient enzyme. So if it is a normal modification or a less efficient modification, enzyme is still there, no. So there is no much of a difference that you will see in the phenotype. But if there is a non-functional enzyme or there is no enzyme at all, then what happens? The other will not produce any kind of a functional enzyme. Okay, so then what happens is whoever is functional becomes the dominant. Whoever is not functioning over there becomes the recessive. Okay, now when there is no dominant gene at all, then there will be no product at all, which we will consider it as the recessive trait. Okay, is that clear? So this modification is what matters when it comes to the dominance. Okay, but now when you look at codominance, okay, codominance is where if there is, so dominance is what? If there are two different types of alleles, only one will express, one should not express. Now here, codominance is nothing but the, for example, in the blood group, if there is A gene and there is B gene, for example, both will be expressed. That is codominance. So in our blood groups, we have IA gene who will code for A type of sugar. So that is the A blood group. Then we have IB sugars that will code for the B type of sugars, which is the B blood group. Okay. And recessive means there is no polymers that are formed. Okay. Now, in case there is an IA and IB present together, both will express themselves, both sugars will be present, and that kind of a blood group is called as a AB blood group. Clear, everybody? But both of these individually are dominant over the I. But both of them, they will be expressed together and they exhibit codominance. So, genotypes can be 6. But phenotypes will be only 4, which is AB, AB or O. Okay. And also the fact that there are 3 genes in the population. Now, the same gene has 3 different alleles, A, B and I in the population, which is called as multiple allelism. This is also another deviation from Mendel's law. Now, pleiotropism is nothing but one gene causing an effect on other features as well. Now, for example, the starch synthesis in pea seeds, it's controlled by one gene. Now, this gene is the same gene who will control the shape of the seed. Okay, so dominant would mean round seed. Okay, recessive would mean wrinkled seed. But in another effect, if there is dominant homozygous, there is a large starch grain produced. Recessive means very small starch grain produced. If it is heterozygous, there is an intermediate starch grain size. So for the starch grain size, it is showing incomplete dominance. But for the seed shape, it is showing dominance. So the same gene is causing 2 different effects. So one gene, 2 effects. Right? This is known as pleiotropism, which is yet again another deviation. Okay, now this is the table for the blood groups, which is there in your textbook. Please do go through it, okay, because it is very important. They'll ask you what is the phenotype, genotype. You may also get crosses based on this, so definitely do go through this. Okay, now coming to the third most important topic, which is sex determination. In sex determination, based on the chromosome pattern, the sex of the child or the organism is going to be decided. So first, Henking was the one who discovered this because he was looking at spermatogenesis and he saw that some cells have some structure, other cells don't have that structure. So he just called it some Mr. X. So X body, like that he called. Later they found out that this was a chromosome and hence called it an X chromosome. Okay. Now those chromosomes that participate in determination of sex are called as sex chromosomes. So the first type is XO type. So female will be XX. Male will be XO. They will have one chromosome less in their genome. Okay. So here what happens is 50% of the sperms will have this X chromosome, 50% will not have the chromosome. So those sperms that will fertilize the egg with the X chromosome will form XX, which is a female. The one with an empty sperm, if it will, empty sperm means without the X chromosome, if it fertilizes the egg, it will form the male. So the entire chromosome number itself will be different. And an example for this is the insects. Okay. Now when you look at XY type, here, the chromosome number will be the same, but just that in the males, there is a different kind of a chromosome that is present, okay, which is the Y chromosome. So, females will have XX, so all the X will have the X chromosome, but the males have XY, so 50% will have the X chromosome and 50% will have the Y chromosome, okay. So, if the X chromosome bearing sperm will fertilize the egg, it will form an XX, a female child. If the Y chromosome carrying sperm will fertilize the egg, it is called as XY, which is the male child. But it is only a matter of chance as to which sperm will fertilize the egg. So there is always a 50% probability of whether it will be a girl child or a boy child. Okay. Now in human beings specifically, 23 pairs of chromosome we have, of which 22 pairs are autosomes, okay, and 1 pair is the sex chromosome. The mother and the father contribute equally 23 number of chromosomes to the zygote. The mother contributes 22 plus X and the father contributes 22 plus X or Y. Based on whether it is X or whether it is Y, it will be a girl child or a boy child, okay. So here there is again, like I said, 50% probability for the sperm to have an X or a Y chromosome. So as a result, there is a probability of 50% to have either of the sexes. Okay. Now the next type is the ZW type. Now so far, males were producing 2 different kinds of gametes. But here, the female produces 2 different kinds of gamete. So male will be producing the same kind of Z chromosomes, okay. Now if it is a ZZ, it is a male. Now the female will have 2 different types of eggs, Z and W. If the Z egg will fertilize the sperm, it will form ZZ, which is a male egg, okay, male bird, whereas W, if fertilized with a Z sperm, it will form ZW, which is a female bird, okay. So like I said, birds are the most common example for the female heterogamety. The last type of sex determination is the haplodiploid. So the total set of chromosome itself is different. Males will have only haploid set, females will have a diploid set. So this haploid set will be producing sperms by the mitosis. The males will be producing the sperms by mitosis. Now the females, they will produce the eggs by meiosis because they have 2 sets of chromosome. Now once the meiosis happens, if the egg will fertilize a sperm and form a fusion product of this fertilization, it will form a diploid female, okay. But if the female will directly produce the male through parthenogenesis without any fertilization, then that is called as the male, because the males will be haploid, no fertilization will take place. If fertilization takes place, it will be a female. So males do not have father, they don't have sons, but they will have grandfathers and they will have grandsons. Understood? So this is called as the haplodiploid sex system. Okay. Now coming to the linkage and crossing over. Chromosomal theory of inheritance was given by Sutton and Boveri. But then there was one scientist called T.H. Morgan who wanted to verify this experimentally. So what did he do? He chose 3 genes in the Drosophila. Okay. So he had body color, eye color and wing size. Okay. Now, when he did a cross between the body color and the eye color, he saw that there was no 9:3:3:1. So, in 9:3:3:1, 9 and 1 is the parental variety, 10/16. 3 and 3 are the new varieties, recombinants that are produced, right? This will be 6 out of 16, which is close to 37%, okay? So, when he did a cross between body color and eye color, he saw that this independent assortment did not take place. He did not see a 37% recombinant. Instead, he saw only 1.3. Majority, 98.7% was the parental variety only. So then he thought, okay, then I should check with another cross. So when he tried another cross, okay, another cross between where is that? Let me just open that. No problem. When he just did another cross between the eye color and the wing nature, okay, wing structure, whether it's normal or miniature wings, he saw that time 37% recombination was there. Means what? These 2 genes are away from each other, so they are able to inherit independently. But these 2 genes are so close, are so linked that they are not getting segregated. They are inherited together. Okay. So higher proportion of parental combination is produced. So stronger linkage means they are together. No recombination is possible. So whenever non-parental gene combinations are produced, we call it as a recombination, okay, which is produced during the crossing over of meiosis, okay. So if the linkage is loose, recombination will be high. If linkage is tight, recombination will be less, okay. So tightly linked genes show very low recombination and loosely linked genes show high recombination frequency. Remember that, okay. So here he saw that the eye color and body color were very tightly linked, but eye color and wing genes were loosely linked and showed the proper recombination. Clear? Now based on this, Alfred Sturtevant, one of Morgan's student, he developed something called the gene mapping, okay. So how do you do that? Based on the recombination frequency, let's say between A and B there is a 17% recombination. That would mean that the genes are 17 centimorgans apart. So 1% recombination is equal to 1 unit, which we call as centimorgan. Okay. Now based on this only, we have done gene sequencing, human genome project, chromosome structure analysis, everything we could do only because of this particular mapping. Clear? Now coming to the last most important topic you need to know in this chapter, it is chromosomal disorders. Okay. So chromosomal disorders will happen because the arrangement or the segregation of chromosomes is not proper, okay. So especially when the chromatid segregation does not happen during cell division, that is when you will see chromosomal disorders. So in that, firstly we have aneuploidy, which means one full chromosome is lost or gained, okay. This is because of non-disjunction, and this is basically trisomy or monosomy we say. So extra one chromosome if it is there, instead of, for example, we have to have one copy from mother, one copy from father, okay. In case there is a 2 copies from father instead of 1, then that means there is 1 chromosome extra, that you call as trisomy. Now same thing, if there is 1 chromosome less, we call it as monosomy. So trisomy and monosomy are the different aneuploidy conditions. Polyploidy means one full entire set only, okay, like instead of being diploid, they are triploid or tetraploid or pentaploid. Now this will be only commonly seen in plants. Animals are not mostly viable in that case, okay. Now when you look at the human chromosome number, we have 46 chromosomes, means 23 pairs. 22 plus 1 sex chromosome from the mother, 22 plus 1 Y chromosome or X chromosome from the father. Okay, so some kind of a sex chromosome from the father. So as a result, we have 22 plus 22, 44 plus XX or XY depending upon the gender. Okay. Now the major chromosomal disorders. Now the Mendelian disorders are completely different. You have autosomal or the sex-linked recessive or dominant disorders there, that you please refer from your textbook, because that is a very, very huge topic. Okay, as it is, this chapter itself is very huge. Okay, I can teach for almost 4 hours with the same chapter, but the problem is when we are revising, that may not be very comfortable. So I am just revising the top 5 topics that you need to remember for your exam. Okay, now coming to the chromosomal disorders, firstly you have to look at Down syndrome. So Down syndrome means the child will be short-statured, round-headed, okay, partially open mouth with a furrowed tongue, okay, there will be a broad palm and one single palm crease will be there. Physical mental development will be hampered and there will be psychomotor retardation that is there. So it is named after the scientist Langdon Down, and here there is a trisomy of 21, means one extra copy of chromosome 21. Okay, so this disease repeatedly asked, so you should know the symptoms and why exactly this is happening. Then Klinefelter syndrome is where there is one extra X chromosome in the male. So instead of XY, there is an XXY. So there will be overall masculine development, but there will be female features like development of breast as well, and the males will be sterile. Now Turner syndrome is where instead of 2 X chromosome, there is only 1 X chromosome and 1 0. Now what happens? 1 X chromosome will start producing the ovaries. Now, if there is another sex chromosome, the ovary will be developed and female secondary sexual characteristics will come out. But if instead of X chromosome, Y chromosome is there, it will tell the X that no, you should not make ovaries, you need to make testes. And the male features and secondary sexual characteristics will come. Now here, since XY both are not there, the ovary started forming, then they didn't know what to do, so it just forms a sterile female with rudimentary ovaries and very poorly developed secondary sexual characteristics. So there is one chromosome less in these individuals. Okay, so with this we are done with our entire principles of inheritance and variation. I hope these 5 topics were useful to you, but remember that in this chapter everything is useful only. Okay, so definitely do read the entire chapter, and to get these notes and to get all our notifications and updates, definitely do scan the QR code and join our WhatsApp community. Okay, our offline crash course is starting March 27th, so definitely do make sure to block your seat because it's filling really fast. And to join our online crash course, you can get this with a 10% off. XPL10 is the code, so you can join now. Classes are starting from December 26th, so definitely do join in. Okay, and with the course, if you want study materials and the tab, you can definitely get it at just 19,000 rupees. So call this number and block your seats, and you can get free offline coaching at our centers for 7 days. So if you would like to take up our free courses, free coaching for 7 days, then also you need to block your seat at the earliest. So visit our Coimbatore, Tiruchi and Tirunelveli centers and get your slots today. So call this number and don't waste any time, because before you know, the NEET exam would be right there. Okay, so I hope this class was useful to all of you. I'll see you all soon in the next class. Take care. Bye-bye.